Hello everybody, and welcome to the presentation of Trenzyme, high-yield production of SARS-CoV-2 proteins from small-scale transfection to GMP compliant production process. And thank you very much to Polyplus for giving us the opportunity to present our data here at the PAX conference. At the beginning, some words about the company, and we are a service-based company providing services for cell line development and protein production. The company was established 20 years ago in 2000. It's privately owned, and we are located in the southern part of Germany. And our clients are usually from the biopharmaceutical industry, biotech companies, but also from research institutes and universities. And we follow high quality management standards, having a QM system in place, and we are ISO 9001 certified since many years now. Our processes are fully documented. We have product identification and full traceability based on a LIMS system, and by use of this LIMS system, we also can offer GMP compliance on request. So I mentioned that we offer different services for cell lines or protein production. This could be either assay cell line development or iPSC differentiation for generating assay platforms, or it could be GMP compliant production cell line development. But more important for the talk today is the green part. That's protein production, or in particular production of SARS-CoV-2 proteins as catalog proteins. All of the projects usually starts with molecular biology steps, and we have a huge collection of expression vectors available, more than 100, and we usually do gene synthesis and clone the targets into our expression vectors. Okay, now let's talk about the virus, the coronavirus. As you know, it's the severe acute respiratory syndrome coronavirus 2. That's the complete name of the virus. It's a positive sense single-stranded RNA virus, and it's most closely related to bat coronaviruses. And there are only 7 coronaviruses known to infect human. One of them, it's SARS-CoV-2. The diameter of the virus is about 100 nanometer, and the genome size is about 30 kilobase per. If we take a look at the structure, then there are some important proteins like the spike protein or spike glycoprotein, or S protein, the membrane protein or N protein, nucleocapsid protein or N protein, together with the RNA. And we have the envelope protein, or E protein, and for sure we also have the viral envelope. If you take a look at the copy numbers, so the spike protein has about 100 trimer copies. That means 300 monomers. The membrane protein and the nucleocapsid protein has each of them about 1,000 copies. And the envelope protein, there are 100 nanomer or 20 pentameric copies present. Now, let's take a look at the spike protein. Here you can see the trimer of the spike protein, and the monomer is given in the ribbon structure, and the monomer can be separated in 2 subdomains, the S1 and the S2 domain. The S1, which is more or less the head of the spike, has the receptor binding domain, or RBD, as most important binding partner, and the RBD is binding to the human counterpart. That's the angiotensin converting enzyme 2, or ACE2. So how do we produce the coronavirus proteins here at Trenzyme, and which proteins do we have? So here's an overview of the proteins we offer. This is mainly the spike protein, the RBD domain in different variants, and we have also a GMP compliant production cell line available to produce the protein under GMP compliant conditions. We produce the spike protein as trimer in different variants. We have the main protease or 3C-like protease in different variants available. We have the human counterpart, the ACE2 protein, and we have the membrane protein and the nucleocapsid protein. And all of them we deliver in very high quality, and we have also cell lines we can deliver off the shelf. On the development, there are further variants of the existing proteins and cell lines, and yeah, now let's take a look on the production process. How do we produce these proteins? Focus is on mammalian production. We have also some proteins produced by E. coli, but here I will focus only on the mammalian production process. So first of all, we select the expression host, in this case, the mammalian cell line. Then we do a gene synthesis with codon optimization and then enter a pilot study by cloning into different expression vectors. And that means we test different secretion signals, we analyze different affinity tags and tag positions, and sometimes we also work with fusion partners. Then we do a small transfection, and this is done at a small scale. That means 3 to 30 milliliter. We use Factor Pro from PolyPlus because it generates very good results, and we produce the proteins and test the purification in the next step. And finally, we finalize the pilot study by having a quality control of the pilot material. After having positive results, we enter large-scale transfection, and here again we use Factor Pro from PolyPlus in the scale of 1 to 18 liter production scale. Also, the purification is upscaled, and final QC will finalize the process, and at the end we have the purified protein available, which then can be delivered to clients. Continuous production we do either by transient transfection. That means we repeat this process here by transient transfection with Factor Pro. This is usually used for scales from 10 to 200 milligrams. But usually we start into a next phase, which is the generation of a stable production cell line. And the stable cell line is usually for scales of 50 milligrams up to 20 grams in our hand. And now let's take a look on the receptor binding domain of the spike proteins. We have different variants available, as mentioned. We have a long version with C-terminal his tag. We have a short version or shorter version with N-terminal his tag. Both of them can be produced by transient transfection or by a stable production cell line. We also have a cleaved protein version available where the his tag is cleaved off by protease. We have bioattenuated proteins available and also bioattenuated receptor binding domain. And we have a tag-free version produced by a GMP compliant process in CHO cells. All the others are produced in HEC293 cells. We have different formulations. Usually we work with liquid formulation, but sometimes we also offer lyophilized formulations, and the production capacity is ranging up to from 2 gram scale per month, depending on the variant. If we take a look to the purification data and activity data, here is a slide showing the purification of the receptor binding domain. It's a quite pure protein. Usually purity is more than 90% or 95%. It runs as a monomer on the size exclusion chromatography, and as you can see here from the glycan analysis, it's heavily glycosylated. If we look into activity data, that means binding data, it binds the human ACE2 very well with an affinity constant of about 1 nanomolar. And even by ELISA, we can determine the binding to ACE2 or confirm the binding to ACE2. And here a comparison of a new batch stored at 4 degrees and an older batch stored for more than 2 months at 4 degrees. it contains the full activity also during storage at 4 degrees. Okay, now some data for the human ACE2 protein. Also, this protein we can purify very nice, with more than 95% purity usually. The activity is perfect. It binds the receptor binding domain. Also, this protein is heavily glycosylated, as you can see here, and it works also well in vivo assays, in binding assays. Here, for example, binding to a hex cell line expressing the full-length trimeric spike protein on the cell surface, and we can use this protein to detect the spike on the cell surface. Now the last version of protein I want to show you is the trimer, and also this protein we get good purity of more than 85% usually. It forms a trimer, as this can be verified by size exclusion chromatography, so no monomers available, only trimer versions. And it binds also the human ACE2 receptor in a similar manner as the receptor binding domain alone. Here in blue, the receptor binding domain alone, and in green, the full-length trimeric spike protein. Okay, so I mentioned that we have a GMP production cell line for the spike protein available, and let's take a look on the process. So first we started with transient transfection because it's fast, it's convenient, but it's non-GMP. And in parallel, we started our GMP-compliant production process by screening different expression constructs, different secretion signals, and then by identification of the best one, we entered the cell line development process, which starts by transfection. Second step is screening of the mini pools. And then after having screened the mini pools by ELISA, we do an upscaling into a fed batch process at about 100 milliliter scale in flasks. After having identified the best mini pools, we do a single cell cloning by image-based determination of clonality. And then next step is again by ELISA selection of the best clones. And the best clones will be expanded, and a second fed batch screen is run to identify the best producing cell clone, which then is available as a research cell bank and can be delivered to clients, or can be used by our company for further development. Now, let's take a look at the results. Here is some titer information. The mini pool ELISA screening round gives usually low titers. Then we see the real titer after having done the fed batch screen of the mini pools, and the fed batch screened mini pools will then be taken to do a single cell cloning, and the clones will be seeded in 96-well plates, and then we screen these clones in the 96-well plates, and the titer is low because it's not a production, it's only grows in 96-well plates. And we select the best clones and do the second fed batch screen in shake flasks for up to 2 weeks, and then we get the final titer. And as you can see here, the final titer is around 1.4 gram per liter. And so at this level, at the mini pool level, we screened around 1,000 mini pools and have done the fed batch with the best 20 mini pools. We screened 250 single cell clones and have done the second fed batch screen with the best 20 clones. And here you see the data from these clones. So most of them produce around, let's say, 100 to 200 milligram per liter, but then we have a significant amount of clones producing around 0.8 to 1 gram, and our top candidate is producing 1.4 gram per liter. To give you a status quo about the process, So we have the stable GMP production cell line available. Our top candidate produces 1.4 gram per liter. We do further optimization, and this hopefully will increase the titer to up to 3 gram per liter. The downstream process is under development and also the formulation. And the GMP production can start as soon as possible. And we use the protein for a very new project. Trenzyme is part of a consortium for a vaccine production. It's called ExpoVax, and the patents are pending, and the animal studies are in progress. So we have a hamster challenge study at the NIH in USA, and a mouse study here at the University in Constance. So I want to show you the big benefits of the ExpoVax platform, and therefore let's talk about the problem of immunization. So all of you know that conventional vaccines, they have a protection gap of about 14 days. It means after the first vaccine injection, it takes about 2 weeks to have a significant antibody titers, and it needs a second injection or a boost to increase the titer. That means during these 14 days, there's a protection gap, and only the innate immune response can protect against infection during this initial 2-week protection gap. And medical staff is needed for immunization. No self-application is possible usually. And even if you get antibodies, it will not protect you from infection and not from shedding. And for sure, no post-exposure prophylaxis is possible. So it's only a prophylactic application makes sense for a usual vaccine. And our solution, it's the intranasal post-exposure vaccine, ExpoVax, which protect people by the innate immunity in the respiratory tract. And this first part of protection then is followed by systemic and mucosal immunity. Since it's a nasal vaccine, the application is easy possible. No medical staff is needed, and it can be sold over the counter. And it's a real vaccine platform, not only for spike S1 from coronavirus, it works also with other target proteins I will show you in a second. But for the spike protein, it's a recombinant version of the viral receptor binding domain, targeting M cells. And the vaccine or the protein has an adjuvant, which is a safe and potent toll-like receptor 3 agonist. And as I mentioned, it's a platform. It's not only a vaccine development for COVID-19. We also started to work on a new target protein. And you may know that the influenza A H1N1 was detected some time ago in China, and this influenza virus could be a future pandemic virus, and therefore we decided also to work on this target protein as future vaccine against influenza A. Yeah, so thank you for your attention, and if there's any question, please feel free to give me a call or to write an email. I'm happy to answer all your questions. Thank you very much.